Well, thank you very much for the invitation to speak to everyone today, and thanks to all of you who decided to not only have your lunch, but hang around afterwards. I wanted to make a nice bright slide to let you know I really don't work for Life Technologies, even though sometimes it feels like it. I'm going to talk a little bit about some of the projects. Some of the projects that were highlighted on a couple of posters presented earlier in the week, and then at the end I'm going to come back and talk about a new NIMH stem cell center that we're participating in, and some work we've done with Life Technologies there as well. So along the way I'll kind of stop and highlight some of the methods we've used, and that leads to some of the products that will be highlighted as well. So our interest in the first project I'm going to talk about is in how restricted precursor cells become different from each other. So in this model, I've taken a pluripotent stem cell and split it into two different kinds of restricted precursor cells, both self-renewable, but each one is pre-programmed to go on and differentiate to some final cell fate. And what's the difference between these somewhat similar looking cells, and how do they maintain that specificity over time? This all started with a project that was begun by my collaborator Marty Grumet and his student Hidong Li, his postdoc Hidong Li. They made a series of induced restricted precursor cell lines from a Fisher transgenic green fluorescent protein rat to make it easier to trace for transplant and so forth. Just by taking cells from a E14.5 cortex, dissociating them, and culturing them, and then infecting them with one IPS-like factor, a VMIC in this case, to immortalize the cells and maintain them dividing so that you can maintain cultures of these. If you grow them in the presence of FGF2, they maintain their restricted precursor stage. If you withdraw the FGF2, they go ahead and undergo a pre-programmed differentiation that seems to be fairly consistent in these cells. So the initial characterization of a series of clones looked exactly like this. Starting out from the lower right over here, this is an mRNA profiling just by a qPCR assay for a series of markers. These markers were chosen specifically because earlier projects had found that some of these cells could be identified as precursors for GABAergic neurons. We were focusing on that fate for this particular screening project. And so some of these markers are specific for GABAergic neuron development. You'll see that they clustered into three distinct groupings, and those three distinct groupings were exemplified by the three boxed cell lines, GE6, GE2, and Cortex-8, CTX-8. These could also be characterized by Westerns for clear markers. Tubulin is a marker of neurogenesis, and GE6 had a strong tubulin marker and none of the GFAP non-neuronal cell marker, whereas GE2 was entirely non-neuronal and Cortex-8 was a mixture of the two, a multi-potential precursor cell. And that can also be visualized here by immunocytochemistry. So the first point of this entire project was to ask about a precursor that was fated to become a GABAergic cell. And so we picked out GE6 and characterized it in detail. And that cell, when co-cultured with hippocampal cells, the GE6 is easily distinguished by its FGF labeling. When that is co-cultured with hippocampal cells, it can be traced for an action potential, and then when cultured by itself and forming synapses on its own cell type, it forms both iPSCs and EPSCs as shown in these traces. And the iPSCs can be reversed by adding bicoculene, indicative of a GABA receptor. So these cells not only become electrically active, and interestingly, they gain action potentials early upon differentiation, but it takes several days more to gain a synaptic activity that's characteristic of these cells. So this was what the project was really after. We went ahead working with Miles Fennell and John Karate at BMS to do an Affymetrix profiling of these cells along with a few others that we'd been studying. And you'll see the kind of trajectory in three-dimensional principal component space here. The small balls represent early time points, the larger balls later time points, and the colors distinguish different cell lines. The neuronal fate is this trajectory up here, whereas the more glial-like cell is in this direction down here, and not surprisingly, the multipotential cell is kind of halfway between the two. If you look in more detail at a hand-selected list of markers that should be corresponding again to a GABAergic developmental profile, you see that GE6 has this very nice progression from a triggering series of transcription factors that are present before differentiation occurs to a second set of transcription factors, including the DLX family, the distal X homeobox family, that gets turned on early upon FGF withdrawal. and then later on markers of neurotransmitters and synaptic proteins that start to come up. And you don't see this kind of progression in either of the other two cell lines. So how are those cells pre-programmed? On the previous slide, I showed you that there was distinct differences in the progression of transcription factors, but at 0 time, the cortex-8 cell actually has a pattern among these genes very much like the GE6. So even though, going backwards. Even though transcription factor patterning is one way to establish different fates on a cell, other ways include microRNAs, which I won't talk about in these particular cells. I'll get to another story with microRNAs later. And the third would be epigenetic marks, and that's where we focus for most of the rest of this project. And certainly, cells in the migratory stream in cortical development can be affected by the soluble growth factors that are present in cortex, and like neural crest, may be affected by cell-cell contact during that migration as well. So what's the default fate, and how is it altered? ChIP-seq shows, very interestingly, that the two cell lines, the multipotential cortex-8 on the left and the GE6 pre-neuronal cell on the right, share very few pro-transcriptional marks. This is the K9, K14 acetyl mark, and very few overlapping K27 trimethyl repressive marks. So it suggests that overall, they actually have very little in common epigenetically, even though they look very similar and they have very similar transcription factor profiles. Now, of course, I'm not showing you how much of this is overlapping poise. I'm just dissecting out the two separately. And if I go one step further with this and look at lists of the top 10 gene ontology groupings that were associated with the transcription start sites near the epigenetic marks, I find that GE6 is currently repressing a whole series of developmental programs unrelated to its fate. So for example, kidney morphogenesis, gland morphogenesis, epithelium markers as well. Whereas the cortex-8 multipotential cell has among its top 10 list several nervous system categories, including neurogenesis. So it looks as though cortex-8 is actively repressing transcription of a program that would lead to neurogenesis. However, and I'll stop and do a commercial right here. When I talk about ChIP-seq, what I'm going to stop and talk about is the fact that we work with, and actually we're lucky to help with beta testing on some of the Magnify and ChIP-seq kits that are available from Life Technologies. Magnify was developed specifically to take advantage of magnetic bead collection of your immunoprecipitated chip complexes, and it works on very small numbers of cells. We've done it on as little as about 1 to 200,000. The spec on the kit is down to about 10,000 cells, which is actually very useful in a lot of stem cell applications. It's fast, has a high yield. We turn through several dozen of these in a day very easily. And all these get run on solid sequencing, and the big advantage for us of solid sequencing has been that over the last year especially, the density has gotten so good and the multiplexing has allowed you to put so many samples on one slide that the cost goes way down. So we're getting a lot of reads for a relatively low cost very quickly because we can mix many ChIP-seq libraries together using multiplexing. And in fact, the advantage here is that the barcoding in solid is read completely separately from the main sequence, so it doesn't take away from your sequence. And I mentioned up here color space sequencing as well. Early on in solid, a lot of folks were kind of afraid of the concept of not using base sequencing and a little concerned about this color space sequencing. As it has turned out, finally, there's really no difference in processing the files. I use Bowtie, an open source academic written software package for genome alignment, and they fully support color space as well as FastQ from Illumina as well. And it's pretty much identical from that point forward. I use the FindPeaks package from Vancouver Short Reads package. And for the Venn diagrams and the gene ontology analysis, I found that, I don't know who wrote this, but someone wrote this beautiful R package called ChIP-beak Anno for summarizing high-level chromatin markings. And actually, I should mention as well that even though we haven't done anything with these yet, we just ordered 2 ion torrents. And for us, the ion torrent is going to be phenomenal because it's fast, it's cheap, and the density isn't up to the level we'd need for ChIP-seq quite yet, but by the time I think we'll be ready with the protocols, the density will be where I want it to be. The lengths of this are amazing, 200 nucleotides in 2 hours. Okay, back to the science. So cortex-8, of course, being a cell that's somewhere in its migratory stream is responding normally to the changing factors that are present in the embryo. And of course, there's this geometry of extracellular factors that's present in the migratory stream. And this cell is, in fact, sensitive to those factors, not surprisingly. You tend to get more glial cells with a CNTF treatment. You get less mature cells with a sonic hedgehog agonist. And you get more mature cells of both cell types with a BUMP2 molecule as well. In addition, several groups had reported that inhibiting histone deacetylases caused more neurogenesis, and we found this to be true as well in this system. So again, since we were interested in the epigenetic markings, we followed that one down and asked whether the valproic acid enhanced acetylation of the histones, which is easily observable on this Western, led to changes in patterns on the chromatin markings. So we went through and data mined the ChIP-seq analysis, and this led down to a very small number of genes, very nicely, that was specifically induced or altered by the VPA treatment. And the pro-transcriptional marking identified a small group of genes that when we looked at the functional categories included, not surprisingly, nervous system development and function. And when you drill down even deeper as to which genes are enclosed within this grouping, you see a number of obvious candidates in here, including things like FOXG1 and some Wnt family members. I highlight specifically NR2F2. This is a protein that binds retinoic acids, and so is involved in some aspects of neurogenesis. And you'll see on the mappings here that without VPA treatment, you see a repressive mark early after FGF withdrawal that slowly gets resolved to a minimal pro-transcriptional mark, whereas with valproic acid, that inhibitory mark is just gone right from the very beginning. And you see a very dense pro-transcriptional acetylated mark here as well. This is confirmed by real-time PCR as well. Back to that pattern of transcription factors in the neurogenic line. If you look at the cortex-8 culture, you see that there is this pattern of transcription factors that's very much like GE6, but nothing happens to it over development. We picked out that among these factors were three of them that seemed like they might be good candidates for mimicking the effect of the GE6 GABAergic cell. ASCL1, about the time that we picked this out, it was about the same time that the Wernick lab published the 3 factors that promote neurogenesis from fibroblasts, the most active of which was ASCL1 or MASH1. DLX1 and 5 have been studied in several systems, including cranial development and cortical development, and knockouts suggest that it was important for some inhibitory neuron development in the brain. So we thought if we took an overexpressed ASCL1 to promote neurogenesis and the DLX family to try to promote pushing toward an inhibitory interneuron, can we change the fate of cortex-8 that's already pre-programmed to be similar to this kind of a precursor? And so we called that treatment ADD, which is kind of an inside joke for the graduate student that works on this. We have little questions about sometimes. If I take what's known about these factors and relate them to the stimulatory growth factors that I already told you about, this is actually my first draft of a map. I literally went to the literature and pulled out the first 5 papers and drew this freehand. There's a wealth of interconnection between these that all lead to neurogenesis from ASCL1 and stimulation of GAD67 and GABA fate down the path from DLXs as well. And I wouldn't put too much stock in the precise connections here. As I say, I just grabbed this in the last week from a couple of papers. So my question now is, if I've got distinct restricted precursors, can I reprogram them with a set of transcription factors that are designed to short-circuit the growth factor stimulation that we know should work in these cells? So certainly just adding MASH1 alone enhances neurogenesis, and this is no surprise. And it actually is quite distinct. What isn't shown here, this is just tubulin staining counting. What isn't shown here is the drastic effect on the morphology of the cell. We've had some discussion about exactly how to quantify that, and we finally found a nice software package to help come up with measures of the shape of the cell, and that's being done this week, and I don't have those results, unfortunately. When we transfect now in all 3 of the transcription factors, you see not only an increase in neurogenesis, but you see this very distinct morphology that's been pretty reproducible in these cells. And these cells now are expressing synaptophysin, a much more mature marker than we ever got with regular cortex-8 by themselves. And we also see what looks like the beginning of a GAD67, 65 marker in a more nuclear location. In untransfected cells, we see more diffuse staining in the cytoplasm. In the transfected cells, it looks more nuclear. But when we really look where the rubber meets the road, the electrophysiology, the results are hugely distinct. The top line is a controlled transfected cortex-8 cell, where you really do have action potentials, and they have a good reversal potential, but they're minuscule compared to the volume of the iPSCs and EPSCs we see in the transfected cortex-8 cells. And so the fact that we see the beginnings of an inhibitory iPSC in here suggests that we may be on our way toward a GABAergic fate. These were co-cultures with hippocampus. They're measuring receipt of synapses. We have to do individual cultures with these cells to prove that they can auto-synapse and have the right phenotype. So that's in progress. Also in progress, these 3 transcription factors are all 3x flag tagged, and I tried to get for you today the ChIP-seq data on where these things are binding. I got the data last night off the solid and started the alignment over last night, didn't finish this morning, so I can't show you the results. Sorry. I will point out, though, that in all these transfections, we're intentionally using plasmids because we don't want these things, 1, to hang around, and 2, we don't want them to integrate. So a plasmid transfection lasts for about 7 days, and we'd previously published a study showing that both Neon, over here on the right, a product of Life Technologies, and Amaxa from a competitor, has good transfection and good viability in both stem cells and in the rat precursor cells that we were studying. And I finally threw in here this as well. We just ordered our new Attune, and we can't wait for it to show up because we've tested it and found that this flow cytometer, using an acoustic focusing method, is much less likely to get clogged by clumpy cells like stem cells. And so we're really excited about using this. And it's reasonably priced compared to the BD instruments we were used to be using. So this ought to be interesting. Okay, ended the commercials. So now let me switch gears a little bit and talk about microRNAs. And I'm going to very quickly review a study that's published and kind of give you the up-to-date details of where this is from current work. I don't have to explain to this audience how important microRNAs in early pluripotentiality, but one of the key points that doesn't get talked about very much is that bioinformatics groups have proposed as many as 10,000 hairpins that are potentially encoding microRNAs in the human genome. And yet we've only found about, let's say, 1,000 to 2,000 of them are currently in miRBase. It's not known what all of these things do and whether they are really just family members that are having parallel functions, but we thought early on that a good way to go after these would be to use a very deep, direct sequencing method. So we did this, let's say, about 2 years ago, took the H1 cell line and compared undifferentiated to NSC differentiated cells. Now, the logic here was that we felt as though early in differentiation is when you would be seeing the most important regulation of microRNAs as these cells are funneling their fates towards specific outcomes. So we sequenced small RNAs in the solid platform. At that point, it was the only way to get strand-specific microRNA sequencing. That has now changed. We aligned the genome. We modeled this to a software model of human hairpin sequences. And then, because we were doing this in kind of a high scale, we couldn't go through one by one and show that they were really biologically active microRNAs. The best shortcut we could think of was to immunoprecipitate for risk complex association. So we had an antibody against Argonaut-2, one of the components of risk complexes, and we did RIP-seq, where we did RNA immunoprecipitation followed by sequencing on solid, and used that to validate the candidates that we had found just by direct sequencing. And we ended up with about 138 microRNAs. These are currently present in miRBase ever since version 15, and here's the reference for that study as well. We found that these were regulated during differentiation. They are found in adult tissues as well. But recently, Eileen Oney, working in my lab, has preliminary results showing that we can detect their biological activity as well. And what she's done is just to design a synthetic luciferase-based clone with a artificial sequence downstream that should be targeted by a novel microRNA. And in this experiment, compared to the uninserted plasmid, she sees a slight reduction that isn't significant with a small number of replicates. but we can do a positive control by overexpressing the hairpin for the novel microRNA to show that the system works and hasn't been saturated. So this will be extended by Eileen as she goes further with her project as well. Finally, we did a nice project with Uma Lakshmipathy of Life Technologies to compare ESC and iPSC microRNA by solid sequencing. And this is just one iPSC line. This is actually a Y cell line that we bought early on before we started making our own iPS lines. And what you'll find, first of all, is that most microRNAs are present essentially at equal contributions in both categories of cells. So the kind of short answer is they're pretty much identical. The more interesting answer, however, is that there's some interesting differences. And the most interesting differences are that the miR371, 372, 373 family are underrepresented in the iPS line. And we went in and hypothesized then that this was due to slightly incomplete reprogramming of that particular cell line. Now that's one cell line, of course. So based on that logic, again, Eileen working in my lab found that she could just pull out 4 TACMAN assays for known microRNAs that were displayed on that previous graph and use them to categorize or classify cell lines that we'd been constructing. So these are some of the very first iPS lines that were constructed by us and our collaborators at Rutgers. And we found that they clustered very nicely with a small number of microRNAs as an easy bioassay, just to show that they're hugely different from the input fibroblasts that they were made from. And she's gone on and shown that she can further distinguish some of these based on minor variations in microRNAs as well. So this might be a very good diagnostic for kind of a quick and dirty use. I'm going to stop at this point and talk about a new development that we're part of. And this is very new. This is a project that only became active June 1. And that's the NIMH Stem Cell Center at RUCDR. Not being a big group of geneticists here, you may not have heard of the RUCDR, the Rutgers University Cell and DNA Repository. This is the largest university-based collection of human genetic material in the world. It has over 250,000 blood samples frozen. We have literally 2 buildings full of liquid nitrogen tanks. Virtually all of them have been SNP ID'd, and most have already served as part of a GWAS. And so there's this full collection of blood sitting there waiting to be used by us to make stem cells. You don't have to go back to the patients, they're fully consented. This is a unique opportunity to take advantage of this incredible genetic repository. So the largest collection from the RUCDR is the NIMH Center for Collaborative Genetic Studies on Mental Disorders, and at a separate location, because this is all de-identified cells, is a full patient diagnosis and history, and so lots of information is available about these cells. So the Stem Cell Center is a newly formed component of RUCDR, which is designed to accept, bank, and distribute source cells for making iPS primarily at this stage of the game, and then to create a small number of iPS lines from some of these cells for distribution to mental health geneticists and researchers. And we just launched a new website literally 2 days ago. And all that's there so far is a fill out this form and request more information. So if you're interested in this, please do go to the website and click on the link. So the interesting point here is that this collection is largely blood, almost there. And I'll do this quickly. The largest component of the collection is blood. We've already made some cells from fibroblasts, and these are characterized using the stem cell TACMAN assay, using a standard TACMAN array to characterize cell lines. But what's really important about these cells is the fact that there are this huge collection of lots of diagnoses already present as bloods, and there are now methods for turning T cells and possibly even EBV transformed lymphocytes into iPS cells. So among those methods is Sendai virus. And we were very excited about trying Sendai virus because it's integration free and it should have a high infectivity for cells like T cells. And we couldn't get our hands on Sendai. It turned out that Life Technologies had licensed it, and so we were lucky to get our hands on it just literally 2 weeks ago and try it out. So I left their logos on the slide just to let you know that these aren't my slides. They're selling 4 tubes containing the 4 factors. And they find that there's a very rapid reprogramming with no genomic integration, and characterization of the lines is pretty much the same as what you'd see anywhere else. So what we did was we followed, following the protocols that had originally been designed for Sendai viruses, we did a very quick infection with 1 shot with Sendai viruses, very preliminary, done the last 2 weeks. And by just PCR of the RNA components back from this, we find Sendai genome, easily detectable at any multiplicity of infection, and all 4 factors present at every multiplicity of infection. When we look by FACS to ask how many cells are OCT4 positive, we actually got a very small number because, again, it was our first try at this infection, but it literally worked the first time. So that's really all I'm going to say about the Sendai virus system at this point. Lastly, we've been lucky to have support from Life Technology for the last several years to have a one-day free stem cell symposium in New Jersey, and we'll be holding that again this September 28th. If you're in the northeast of the United States, please come visit us on September 28th. And this is co-sponsored by an NSF training program as well. And with that, I'll finish up and thank the folks that contributed to all the work. And I want to specifically point out Jen Moore, who is our head of stem cells in our lab and is really a magician at working with these cells, and Mavis Swerdell, who's made all of our solid sequencing libraries as key people. And then finally, Chris Recupero, who did all that chip work that I talked about, and Eileen I already mentioned, and various collaborators and financial support. With that, thank you very much.